Hello everyone, welcome to the Wise Studio YouTube channel. Today's video is video number 4 about our second macromolecule, lipids. So lipids have a wide variety of functions in biological systems. The first one being energy storage. So whenever you're, we learned in the previous video that glucose and your carbohydrates are really your quick energy sources. Well, lipids are kind of the backup of that. They are an energy storage. So whenever you run out of glucose and simple sugars, you can always rely on lipids and burn the lipids for energy. Cell membrane composition. Here's a picture of a cell membrane, or a diagram, I should say. And each one of these blue dots is what we call a phospholipid, which is a special type of lipid that makes up the cell membrane. And we'll talk about that in more depth in the coming slides. Another function, an important function, is nerve cell insulation. So we've got this structure, which is a nerve cell or a neuron, and we've got this structure called the myelin sheath. The myelin sheath is a fatty, like a lipid insulator that allows action potentials to travel from each junction there. And we'll talk about action potential later in the course, but just know that it does play a huge role, and the myelin sheath plays a huge role in action potential and actually facilitating the process. Finally, it does protect organs by lining certain organs, especially your digestive system organs are lined with a lot of fat, and that is for protection. So the monomer, or I should say two monomers, there are actually two components of lipids, or of the bigger polymers of lipids, including fatty acids. So usually there are multiple fatty acids and one glycerol backbone, and we'll see those structures in the next slide. For polymers, we've got diglycerides and triglycerides. Obviously, you could see the prefixes there, di meaning 2 and tri meaning 3. And when we're talking about the number 2 or 3, we're talking about 2 fatty acids or 3 fatty acids. The glycerol backbone is most of the time, 100% of the time that I've seen, only one glycerol backbone is found in a lipid, in a monomer. And so the next really important thing to know is the bond type. And when fatty acids are kind of attached to the glycerol backbone, they create this ester linkage, which we'll see on the next slide. So here is a picture of the triglyceride structure. We have the glycerol backbone right here, obviously unattached from anything. We've got a free fatty acid molecule right here. Whoops. A free fatty acid molecule right here. And now we've got a triglyceride. This entire thing is a triglyceride in which we've got the 3 fatty acid chains as well as the glycerol backbone. Now, the red arrows are pointing to what we call the ester linkages. The ester linkages obviously link the glycerol backbone to the fatty acid chains. And so you've seen this is one of the bonds, this is one of the linkages or bonds, and this is one of the bonds. And the other thing to notice about the free fatty acids and the fatty acid structure in general is we've got this carbon chain. And we'll see in the next slide some differences that could occur within these fatty acid structures. The next thing that we're going to discuss is saturated and unsaturated fats. And so what you need to know about saturated fats is that they're straight in structure. They've got this carbon backbone, and they've got hydrogens attached to that usually, and they're pretty much straight. And that is because they are saturated with hydrogen atoms. So you see every place where there is a hydrogen atom possible or placed, you'll see a hydrogen atom. And so there is no presence of double bonds, at least in the carbon chain itself. And another tendency of the saturated fats is to be solid at room temperature. Unsaturated fats are the exact opposite. Okay, oftentimes they are bent. And that is because they are not fully saturated with hydrogen. You see the presence of double bonds within it. It could be at one double bond or two or more. Basically, you could see how that this double bond takes away two hydrogen, takes away the spot of two hydrogen atoms. And you could see, thus, it's called unsaturated because it's missing two hydrogen atoms that it would be saturated with above. And those tend to be liquid at room temperature. There is, however, another distinction that I want to make between monounsaturated fats and polyunsaturated fats. Again, the use of prefixes is really important in this unit. You see mono meaning one, and that is because they only contain one double bond. Poly meaning more than one generally, and that is because they contain more than one double bond. So just something intuitively that you'll need to know with the distinction between monounsaturated and polyunsaturated fats. But usually the main difference, at least molecularly, is that the unsaturated fats tend to be bent. Phospholipids. Now, phospholipids, as we've kind of started to discuss on the first slide, are specialized types of fat molecules or lipid molecules. And so what happens is they're actually the ones that construct the cell membrane. We call a cell membrane, another name for it is a phospholipid bilayer. There is bi, again, a prefix meaning two. So there are two layers of phospholipids, right? And so if you want to take a look at the specific structure of a phospholipid, you'll see that there's one normal saturated, fully saturated, as you can see, fatty acid. And then we've got a bent one. The bent one is obviously due to this double bond right here, and we call that an unsaturated fatty acid. So these two these two fatty acids, are obviously attached to the glycerol backbone that we talked about. So everything from here on down, if you draw a line here, you would basically call this a diglyceride, which we introduced all the way back here, diglyceride. And that is because there are two fatty acids. But there's an additional structure at the end of the glycerol backbone. We see a phosphate group, and we'll talk about more about functional groups in our next video about proteins. But the phosphate group and this choline group, it's another functional group, these make this head of the molecule polar. They are they make an alcoholic compound that is polar. However, we've got nonpolar tails. You can see that they're nonpolar, and so these nonpolar tails are actually facing on the inside, while the polar heads face on the outside. And so molecules with both hydrophilic polar and hydrophobic nonpolar components are called amphipathic. So phospholipids, are called amphipathic molecules or compounds. And the reason why they arrange in this certain configuration, we will discuss more in the cells unit, the upcoming cells unit. But basically, in the cell membrane, not in the cell membrane, but outside of the cell membrane, you've got polar molecules on both sides in the form of cytoplasm on the inside of the cell, and liquid and water and other polar molecules on the outside. So your nonpolar tails, which another word for that is hydrophobic, don't want to be near water. They fear water. So because they don't want to be near water, they fold in among themselves right in the center. The polar heads love water. They don't really care about avoiding it. They love it. So they're going to be on the outsides, and you're going to see a configuration like this making up the cell membrane. So there are other examples of fats that we see. Carotenoids are light-absorbing pigments. You've got steroids, which have shared carbon rings and are highly hydrophobic. Vitamins, which are small molecules that you must acquire in a diet. They are not synthesized in the body. And you've heard of all the vitamins, vitamin D, vitamin C, all of those. And waxes. Waxes are highly hydrophobic. And I think you guys know that to be true from real life as well. And so these are just some of their structures. I would say the use of this slide is to try to memorize or know, at least it is good to know, at least one of these as a potential other example of a fat or a lipid. You need to know phospholipid, you need to know saturated and unsaturated fats, and you need to know the general triglyceride. But here you've got four other types, and I do recommend, though at least the one that I tend to gravitate towards or recommend, is the steroids. They have shared carbon rings. You see that here in the steroid called testosterone. You see the shared carbon rings, and they are highly hydrophobic. Those would be the only two details you would need to know. Here is a wax, and you see this long carbon chain similar to a fatty acid chain. Um, and that is highly hydrophobic in itself. You see in the bottom corner there, here are vitamins. Vitamins are kind of a mixture between the two. They've got some of these carbon chains along with carbon rings, all right? And then you see finally your carotenoids, which are very similar to this wax structure in that they have these carbon, this long carbon chain, but they do have some types of carotenoids also contain these rings. And so I hope you enjoyed this video and I tell you a bit more about lipids. Our next video will be diving into proteins, which is a bit more involved, and I'll see you guys then.